Okay, class, now we're going to take a deeper dive into the cells and look at all the little smaller components, the subcellular organelles and structures within the living cell. This next slide will be an important one because it summarizes the things that we're going to be talking about. I have on the left a list of other various subcellular organelles structures, and on the right is a sort of a generalized depiction of a typical eukaryotic cell. Now, this would be an animal cell that does not contain chloroplast, which would only be found in plants and photosynthetic algae. But we'll go through this various list of subcellular organelles and tell you a little bit about the function of each of these. Now, before going on, let's just point out that prokaryotic cells are simpler in structure. They do not have a well-defined nucleus, as do eukaryotic cells, and also they will not contain mitochondria. But first, let's talk about the nucleus, which is usually the largest subcellular organelle in a cell. It's where the DNA is located, and we call the DNA chromosomes when it's in a stage where it's more defined in its structure, as shown by the bow tie looking chromosome structures in the diagram. When the DNA is less structured, that is, it's not as defined as the bow tie structures, we call it chromatin. But the DNA is located inside of the nucleus, and the nucleus has a double membrane, one of these bilayer membranes, which surrounds all of the DNA, all of the chromosomes or chromatin. The DNA, of course, contains the genetic information. It is the blueprint for making all of the proteins that we have in our bodies, and we'll talk more about that in a different chapter. The DNA information will have to be transcribed to messenger RNA, and then the messenger RNA will have to be translated at the ribosome sites, which we'll get to shortly, to make a sequence of amino acids, which we call proteins. Also, when a cell divides, all of the genetic information, all of the chromosomes have to be replicated. We'll talk about that in a process called mitosis. Now, the second subcellular area, it's not really an organelle, is the cytoplasm, which basically is the rest of the stuff. That's not one of the other subcellular organelles that we're going to talk about. It is, you might consider it to be the soup, or at least the broth of the soup inside of a cell, and it's where all the other materials are dissolved, the various ions and molecules, things like ATP, and it's where the messenger RNA swims through to get to the ribosomes, etc. So you have the cytoplasm, all the other stuff within inside of a cell. Another important structure to point out is the endoplasmic reticulum. Now, if you ever play this game called Hangman, where you come up with a word that's difficult for other people to guess the letters out, these are a couple of good choices: endoplasmic and reticulum, to use in your Hangman game. But the endoplasmic reticulum is a network of membranes that sort of extends out from the nucleus. And as you see in the diagram, if you first look at the nucleus, the center part, then it's sort of like concentric layers, almost like the layers of an onion skin that sort of grow out from the nucleus. And sometimes this endoplasmic reticulum is called rough endoplasmic reticulum, and sometimes it's called smooth. The rough endoplasmic reticulum is the site for ribosomes. That's what the rough part is. It's where the ribosomes are located. And the ribosomes are the site of protein synthesis. It's where the messenger RNA travels from the nucleus to the ribosomes in the cytoplasm part of the cell. And it's the site at which the amino acids are strung together using the information, the genetic information carried on the messenger RNA, to make the proteins, which are the functional molecules of our body. This process of synthesizing proteins requires energy, and the primary source of that is something called GTP, which, as I indicate here, is sort of like a sister or a brother of ATP. Two other important subcellular organelles are mitochondria, which we find only in eukaryotic cells, and chloroplast. Now, both of these have to do with energy transduction. That's why I've grouped them together. Mitochondria are more or less sausage-shaped organelles. You might think of them as sort of like the furnaces of a cell. It's where the fuel, the final stages in which the fuel is burned up to extract energy. The fuel molecules, the most important one we'll talk about a little later, is glucose. We will see that glucose is metabolized to form ATP molecules, these sort of energy currency molecules that go around and provide the energy for other processes within a cell. So think of mitochondria as the sort of furnace of a cell. The latter stages in the metabolism steps that occur in mitochondria involve oxygen, which is sort of important. The other stages occur in the cytoplasm, in the more aqueous phase parts of the cell. But the final steps occur within mitochondria, and these are the oxygen-involving steps. That is, when we breathe in oxygen, it's basically because these processes occur within our mitochondria to extract energy or to obtain energy from our fuel. If we are a plant or a algae or some photosynthetic bacteria, we would also have another subcellular organelle called chloroplast. Now, usually we think of these as being green because they contain this important pigment molecule called chlorophyll. The cytoplasts are the location of photosynthesis, this extremely important process that occurs on planet Earth, where energy from sunlight converts carbon dioxide and water to glucose, this fuel molecule, and oxygen which we have to breathe. Basically, this process is why we can live on planet Earth. There's an interesting theory about both chloroplast and mitochondria, because it appears that through an evolutionary process, both mitochondria and chloroplasts were captured by cells to become the modern eukaryotic cells and plant and algae cells. And this is called the endosymbiotic theory, which states that chloroplasts and mitochondria actually originated as their own individual bacteria that were engulfed by larger cells to become eukaryotic cells. That is, imagine that there was some universal ancestor prokaryotic cell, and that one stage of the evolution is that the DNA in this universal ancestor prokaryotic cell developed a nuclear membrane. That's why eukaryotic cells have a nuclear membrane. And that as time went on, some other of these universal prokaryotic ancestor cells developed this amazing ability of photosynthesis, and others developed this ability to metabolize using oxygen, so that these bacteria became the precursors of what are now chloroplast and mitochondria, so that the mitochondria and the chloroplast became symbiotically engulfed into this cell to lead to what is now the modern versions of eukaryotic cells. Now, there's evidence for this theory includes the fact that both chloroplast and mitochondria have their own DNA. That is, in addition to the DNA that is in the nucleus of a cell, the mitochondria and chloroplasts have their own DNA, and it is circular, whereas the DNA in the nuclear region is usually linear. Now, we're getting into the weeds here about what's circular and linear, but it's an important distinction. Also, there are separate ribosomes in chloroplast and mitochondria that are different from the ribosomes in the rest of the cell, and the way in which the process begins, the first amino acid formed in the synthesis of proteins is different for proteins synthesized in mitochondria and in chloroplasts compared to all the other proteins. This table sort of summarizes these important differences. There are a few other entries in the table. It's not necessarily for us to go into this, but the point is that there's very good reason to believe that in modern eukaryotic cells evolved as a result of this symbiotic relationship of chloroplast and mitochondria into becoming engulfed into cells to become these modern eukaryotic cells. Now, some other subcellular components include, and these are, we're getting down into the last entries in our table, but Golgi bodies, lysosomes, vacuoles, and vesicles. Golgi bodies are what is sometimes called the Golgi apparatus. Is a site where synthesized proteins that are synthesized on ribosomes, are further processed, where you add some other parts or components to the structure of proteins, or where they're processed for being exported outside of the cell. Lysosomes, if you kind of think of lysosome, Lysol, is kind of like a disinfection site. That is, it's where some of the enzymes that are worn out are broken down as cellular waste. So lysosomes, think of Lysol, or disinfecting or getting rid of the waste components within a cell. Vacuoles are storage sites for waste materials before they're expelled out of the cell. And vesicles is another type of subcellular component that has a variety of different functions depending upon the cell. In one example, in nerve cells, neurotransmitters are sort of stored inside of vesicles before they are released during the signaling between nerve cells. So vesicles have different functions, but they're sort of components, or you can kind of think of them as backpacks or gym bags, and where different things are made and stored before they're released. And lastly, let's talk about the cytoskeletons. Cytoskeleton are the sort of girders within a cell that are long filamentous protein structures that sort of give some kind of internal support or structure to the cells. They can also be involved in transporting vesicles or proteins from one place to another within a cell, or in some cases for the shortening and contracting of cells. That's why some cells are able to sort of inch along or move or wiggle, because of movements of the cytoskeleton within cells. A couple of more familiar ones of these are actin and tubulin. Now, you've probably not heard of tubulin. Sounds like a tube. But actin you may have heard of because it's involved in muscle cells, actin and myosin inside muscle cells. So actin is you can imagine it's a filament that is involved in sort of contraction and expansion of both the cell itself and maybe moving things within the cell. Okay, so those are the important subcellular components in a typical living cell. We'll pause again, and when we come back, we'll talk about energy transduction and replication or reproduction of the living cell. Okay? See you shortly.